Pharmacogenomics, often abbreviated as PGx, is the study of how genes affect a person's response to medicines. PGx testing is the analysis of genetic variation in genes associated with medication response, such as pharmacokinetic and pharmacodynamic effects. Pharmacogenomics data, along with other clinical information, can help inform medication selection and/or dosing, and improves outcomes for certain medications. Recognition of the importance of genomics as it relates to medicines dates back to the 1950s. However, the completion of the Human Genome Project in 2003 provided a reference genome. This advancement catalyzed the field, and researchers have made enormous strides in identifying, predicting, and validating the effect of genomic variants on outcomes for certain medications. One source of difference in how patients respond to medications is a change or changes in DNA that influence how the body processes medicine. Many drug targets metabolizing and transporter proteins are known to be encoded by genes that are highly polymorphic, which means genetic variation. These proteins include liver enzymes, important to the metabolism of many medications, and transporter proteins that carry medicines across biological membranes. Genomic variation in these proteins may lead to differences in concentration of the medication within the blood, due to differences in absorption, distribution, metabolism, or excretion of the medication. The specific pharmacology of the medication influences how differences in DNA can result in different expressions of that medication's effect. For example, from a pharmacokinetics perspective, if DNA differences produce an impairment in an enzyme that's primarily responsible for converting a parent active medication compound to an inactive metabolite, higher concentrations of the active medication would be observed. On the other hand, if DNA differences produce an impairment in an enzyme that's primarily responsible for converting an inactive parent medication like a prodrug to an active metabolite, then low concentration of the active metabolite would be observed. Therefore, the expression of the clinical impact of differences in DNA on medication is dependent upon and specific to the medication of interest. When DNA differences result in changes in how the body responds to or processes a medication, it's called a drug-gene interaction. Just like drug-to-drug interactions that can often be avoided by adjusting medication dose or changing medications altogether, certain drug-gene interactions can be mitigated similarly. Therefore, knowledge of a patient's pharmacogenomics profile can not only inform the likelihood of efficacy and/or adverse events to certain medications, but in some cases can lead to specific, actionable recommendations to improve medication-related outcomes. As researchers identify more genes important in drug response and the multitude of factors that can inform prediction of drug response, the performance of pharmacogenomics is ever apparent. We anticipate that every field will use pharmacogenomics tests routinely, as genetic mechanisms of medication response and disease are better understood. Thank you for your time and attention throughout this video. We hope you found this information to be of value to you and your clinic.